Hello, my name is Sam. I am joined with my colleague Fariha, and today we are going to be discussing fast, accurate, and cost-effective AAV characterization. Adeno-associated viruses are commonly investigated gene delivery vehicles for rare genetic disorders. There are currently 3 FDA-approved AAV products, and there are many, many, many more in clinical trials. On the top right of this slide, you can see this is actually from 2019, so it's a little out of date, but even in 2019, there were over 100 different products in clinical trials. On the bottom, you can see the 3 that are approved: Hemgenix, Luxturna, and Zolgensma. A little background on what an AAV is. AAV stands for adeno-associated virus. It is a non-enveloped, single-stranded virus that belongs in the Dependovirus genus. It is dependent on co-infection with other viruses to replicate, which makes wild-type AAVs non-pathogenic. There are at least 12 different human serotypes, which differ in their tropism, meaning which tissues they infect. Wild-type AAV has a 4.7 kilobase genome containing 2 open reading frames, which have 3 genes: the rep gene, the cap gene, and the assembly activation protein. This is flanked by inverted terminal repeats. The capsid is composed of 3 viral proteins, VP1, 2, and 3, that are present in a 1:1:10 ratio. These proteins are organized in an icosahedral geometry in a 25 nanometer diameter. During AAV production, several impurities can be formed. These include things like aggregates and partially filled or empty capsids, which do not contain any genetic material. On the left here, you'll see the ratio of the capsids and aggregates that are given at the time of harvest and after purification. Empty and full capsids differ in a few key ways. One is isoelectric point, also molecular weight, and their UV 260-280 absorbance ratio. This has led to the development of several techniques that can separate and analyze empty and full capsids.
Thanks, Sam. Thanks for that introduction on AAVs. I wanted to talk a little bit about the characterization of these samples. So let me get my laser pointer here. And so here you can see an AAV capsid. And so here's your protein shell with your genomic material on the inside. And so towards characterizing these materials, there's a few pieces of key information that scientists are interested in. And that's basically those quality attributes that define the AAV. So the first aspect is the genomic DNA. You want to make sure that DNA is actually inserted within that capsid itself. We care about the genomic titer, so how much DNA we actually have encapsulated. On the other hand, we're also curious about the capsid itself and being able to characterize that. I would say one of the most important aspects of that is the empty full composition. So how much of your DNA is within the capsid. We talk a little bit about partial DNA. You see partial DNA up here with the genomic DNA characterization. And partial DNA is kind of this catch-all phrase, which can include, partial sequences of the intended genomic sequence. However, it can include other things like host cell DNA, basically just unwanted DNA within the capsid. But effectively, we're trying to characterize this capsid. We want to know the size of this. One of the things that we see come up sometimes is aggregation of the capsid, whether it's aggregation of both empty and full capsids as well as partials, but that aggregation can impact the infectivity or transduction of the AAV material for further studies down the line. And so kind of showing you a few of the characterization methods currently implemented in labs. I've listed, this large table right here with quite a few methods. And these methods are really developed to kind of characterize some of those aspects discussed on the last slide. And so one of the most common methods that I see that folks use is analytical ultracentrifugation. I would say to many, this is considered a gold standard of AAV analysis for a couple of reasons. One of the reasons we can get an empty full composition on the AAV, it gives us an analysis about other aspects, including aggregates as well as partial analysis. So say you have that unwanted impurity DNA within your AAV capsid. But I would say one of the issues that comes up with a lot of these methods of characterization is definitely the aspect of throughput. So I would say AUC is in this classification. It does not have a particularly high throughput. Sam is going to discuss this in a little bit more detail later on. But what we see is that it often requires a large amount of sample. One single run takes a very long amount of time. And so if you have a lot of AAV samples that you're trying to characterize, you're not really going to get that turnaround time. The same thing can be said for CDMS, charge density mass spectrometry. So like you can see, there's quite a few methods, but one of the big issues is really the requiring of specialized techniques, expensive equipment, and then throughput. Going down this list, talking a little bit about some of our light scattering techniques, we have DLS also as an option. DLS typically is used for bulk measurements, so it's not going to be typically used as a detector that's hooked up with, say, your chromatography, your HPLC system, but it can give you information about sizing, but it's limited in being able to give you information about, say, your genomic material, your capsid, or your empty full composition. One of the things we really want to highlight today is the potential of MALS, multi-angle light scattering. We see MALS detectors very often hooked up in conjunction with some type of LC, typically SEC size exclusion chromatography, which I will be highlighting in just a second. The nice thing about using LC MALS is that we can get a wide array of information, including empty full composition, information about both the capsid as well as genomic titer, information about aggregates, partials, and size analysis. I put a couple asterisks right here and here. Your ability to get aggregate and partial analysis, it very much depends on your mode of chromatography as to whether you get that type of separation or not. So there's definitely quite a few options, but we're excited to give you a little bit more on the LC MALS in this presentation. And so going directly into SEC or size exclusion chromatography coupled with MALS, multi-angle light scattering detection. And so for those of you who might not be familiar with MALS, MALS is an excellent tool to get information about molecular weight. So when it comes to the principles of MALS, the idea is that you shine light onto your molecule. So you can imagine this to be your AAV capsid right here, and then it scatters light in all directions. And so the key piece of information that we're concerned with is how much light is scattered in the forward direction. Based on that value, this directly correlates to the molecular weight of the molecule. And so we're using molecular weight or our understanding of molecular weight to characterize some of those few key pieces of information, such as empty full composition, as well as the titer or concentration of those AAVs. The other important aspect, and this doesn't always come up, it depends what type of analysis you're trying to do, but you can also get information about the size of the molecule. So if you're specifically trying to understand how much of your AAV is monomeric versus aggregated, getting the size of the molecule is really important. And the way that we determine size is what we look at is how much light is scattered in all directions. And we get a ratio of how much light is scattered in the forward direction compared to the other angles. And this angular dependency is directly related to the size of the molecule. However, today we're going to be focused a little bit on the molecular weight. And to show you just what MALS looks like in practice, here is an AAV5 sample analyzed in our lab. So we're highlighting a few things here. The first is we're using our TSK gel GMPWXL column. So this is a mixed bed column. One of the advantages of mixed bed columns is they have a wide range of pore sizes. And so whether you have your AAV monomer in there, maybe you have fragments or DNA that may be leaked out, for example, or you have aggregated material, it's really going to encompass a wide range of sizes. And so maybe you don't know quite what the size or molecular weight of your material. Mixed bed columns are a very, very good starting point. And so here you can see this AAV5 material. And so this particular sample is actually empty, meaning, there's no genomic material on the inside. And we can see the response by light scattering. We get a nice large response from light scattering. And so one of our preferences towards using light scattering as opposed to other methods of detection is that light scattering is not, the signal is not only proportional to the concentration of your sample, but also molecular weight. So the molecular weight of these capsids is quite large, and that's why you see this nice big response. And so from that, we're really getting, that high sensitivity that we want using our MALS detector. And ultimately, we can use that response to get information about that full empty ratio, as well as information about potential impurities, such as, those aggregates or DNA that's not within the capsid itself. And so this kind of highlights that kind of same point. Here we can see the UV response in comparison to the MALS information. And so here are the three angles that we have on our MALS detector. So we have a right angle at 90 degrees, we have a low angle at 10 degrees, and a high angle at 170 degrees. And you can see with all three of these angles, we get a very, very nice response to the AAVs. And so this kind of shows what I was explaining in the last slide, that the light scattering response is not only proportional to concentration, but also the molecular weight of the sample. On the other hand, when it comes to UV is what we consider a concentration detector. So your response is only proportional to concentration. And so unfortunately, with a lot of these, what I put in the gene therapy family, so whether it's AAVs or LNPs or any kind of particle that's encapsulating something, a lot of times the concentration or titer is quite low. And so this is where UV starts to fail. We just simply don't see a response by UV because the concentration is too low. Fortunately, you have light scattering as an additional detector to get more information. And so towards that, we did a few studies to understand how MALS really can be used to characterize AAVs. And the first thing was using TOSO's MALS detector. So TOSO has a MALS detector called the Lens 3. And so we use our Lens 3 detector to characterize the sensitivity of it towards detecting AAVs at a wide range of concentrations as well as titers. And so on the left-hand side, you can see the data from the right angle, and you can see a few different peaks from low concentration all the way up to high concentration. And so visually, you can see a nice response or change in response based on the concentration. And again, we can plot that to get a little bit more quantitative on the right-hand side. And so on the y-axis, this is the response from light scattering. And then on the x-axis, we have this as a function of the titer itself. And you can see a nice linear response from low to high, giving us, really a nice range to work with to characterize AAVs. We work with a variety of types of lab, whether they're analytical labs, process labs, and a lot of times when labs are running AAV samples, the concentration really, there's a very wide range. And so you can see here, we can easily do the analysis regardless of if it's low or high titer. And so kind of turning back to what we were talking about with empty full composition, I would say, in my opinion, this is one of the most important questions is, right, you're making these capsids, you're trying to put some type of genomic information within the capsid. And at the end of the day, we want to only produce 100% full capsids. But as you know, and I'm sure both on our side as well as those watching, we know that there's a lot of things that are kind of classified within the impurity profile, whether those are empty capsid, partial capsids, maybe we have other capsid with host cell DNA, for example. We want to actually be able to quantify that. And that's one of the things that we're really working on with TOSO, and this seems to be a large interest within the industry itself. So on this slide, we are showing the empty full composition analysis. So this is similar to the last slide where I was showing the sensitivity data. And so instead of changing how much of the titer is within the sample of analysis, what we're changing is how much of the sample is full versus empty. And so what we have are samples that are completely empty and samples that are completely full, and we mix them in varying ratios from 0% all the way up to 100%. And so what you see right here are the chromatograms from the RALS, the right angle light scattering signal. And again, we can plot that on the right-hand side to actually kind of get some quantitative information about these chromatograms. And so we see on the y-axis, this is the signal from light scattering plotted as a function of how much of those capsids contain the intended genomic sequence. And the nice thing here is we get a nice linear response from 0% full all the way up to 100% full. I always like to make the comment here that we are not using this as a standard calibration. We're not using a calibration to determine the empty full. Really, this is just a proof of concept that light scattering can be very useful for determining empty full composition. What we are actually doing instead is you'll see in the future of our software a tool to use light scattering response directly to determine empty full composition with a singular chromatogram. It will not require 0% full all the way up to 100% full. It will just require a single chromatogram. But this linearity really shows that we'll be able to do that, and you'll see that in the future of our software. Next, we have Sam. He's going to talk to you a little bit about using a different mode of chromatography. Here we have anion exchange chromatography to analyze AAVs.
All right, thank you, Fariha. So as Fariha already mentioned, there are several techniques that people have used to try and characterize empty and full capsids. She talked a little bit about analytical ultracentrifugation and how it's considered kind of the gold standard, but it has a few drawbacks. So on the table on the right, you can see analytical ultracentrifugation at the top compared to things like anion exchange chromatography, cation exchange chromatography, size exclusion chromatography. One of the major drawbacks for analytical ultracentrifugation is the process time. At the bottom of this page, I took from Strobel et al.'s paper an example of a purification scheme they made for AAVs that uses analytical centrifugation with a cesium chloride gradient. You can see highlighted in the red square, that single step takes 23 hours. I've read a few other papers where it takes, only 15 hours, but generally, if you're going to use centrifugation, your process is going to take over 10 hours. And as Fariha also mentioned, it's not easily scalable. If you're getting a lot of samples, or if you want to do any kind of process scale, separation of empty and fulls, it gets very difficult. And there's also a high potential of lot-to-lot variation due to operator error. There's a lot of room for people's different expertise levels to cause variation when you're doing one run versus another. Anion exchange chromatography is an attractive alternative because it's much faster, it's much more easily scalable, and it has lower potential lot-to-lot variation. So I'm going to talk a little bit about the methods and the materials we use at Toso to do our empty and full analysis using the TSK Gel QStat resin. This resin uses a quaternary amino methyl ligand on a non-porous stationary phase. The fact that it's non-porous is beneficial to AAV analysis because it lacks pores, so it's not going to trap any molecules, it's not going to slow your separation process. It gives you a faster mass transfer, a higher resolution, and a higher efficiency. I'm also going to show you how we use a mobile phase with pH 9, which is above the PI of the capsid, so it sticks to our resin nice and good. We have also investigated several salts, including some non-traditional salts, which offer improvements over sodium chloride. And we have optimized gradients to give you really good resolution, and how we can use fluorescence detection to minimize our sample needs. So some of the salts that we've looked at have included sodium chloride and sodium phosphate and magnesium. We've had very good success with tetramethyl ammonium chloride. If you look in the literature, a lot of people will use this. It is believed to work well because it has a quaternary amine structure, which is very similar to the ligand found on TOSO-Q resins. So if you look on the right at the top, you can see tetramethyl ammonium chloride molecule. And below that, you can see kind of a generic representation of a TOSO TSK gel resin. And you can notice in both cases, we have this amine structure, which looks very similar. So on this slide, much like what Fariha did in her experiments, we used a mixture of standards where we had 100% full, we had 100% empty, and then a 50% empty, 50% full mixture. In this experiment, we were looking at AAV5 in particular. We used a tetramethyl ammonium chloride B buffer at a 1 molar concentration, and we used a 10 to 30% gradient. You can see here in the middle, in the 50% full, 50% empty mixture, we get a resolution of 1.5, which is pretty good. It allows us to get pretty good separation of empty and full capsids. We also wanted to test the limit of detection with the fluorescence detector. So using that 50-50 mixture, we essentially made a serial dilution, and we plotted the peak area versus the particles injected onto the column. And you can see we have on the right, we have a really great linear response. Our R squared is 0.999. So using this, we can determine our limit of detection for AAV5 is 6e to the 9th, and we have a very linear response from 10 to the 9th to 10 to the 11th viral genomes. In this experiment, again, similar to what Fariha did, we used a mixture of different amounts of empty and full standard to create kind of like a standard curve. First, we start with 100% empty. Then we do 75% empty standard, 25% full, 50% empty, 50% full, 25 empty, 75 full, and 100% full. And on the right, we plot these where it's kind of the percentage of empty versus the peak area. And in green, we have full, and in red, we have empty. And you can see in each case, we have a linear response with an R squared, either at 0.99 or very close to it. We repeated this experiment with AAV8, and we got, again, very nice linear responses for both empty and full. Some exciting news we have is that we've developed a proprietary buffer that has two major advantages over tetramethyl ammonium chloride. First, we have improved resolution, and second, we have virtually no toxicity or safety concerns. For anyone who's worked with tetramethyl ammonium chloride, I'm sure you know is pretty toxic. Our buffer is not toxic at all. I'm afraid I can't tell you the specifics of what's in it, but I can show a little bit of what we're doing with it right now. On this slide, you can see 4 different serotypes of AAVs, 2, 6, 8, and 5. Running a 10 to 30% gradient using our proprietary buffer, we did the same kind of separation experience we did before, where we have 100% empty standard, 100% full, and a 50-50 mixture. Below each chromatogram, you can see the resolution of the 50-50 mixture, and you can see in all cases, the resolution is above 1.5. For AAV6, we get all the way up to 2.4. Directly comparing our buffer with tetramethyl ammonium chloride buffer, on the bottom left, you can see the little table. We can see for AAV8, the tetramethyl ammonium chloride gives a resolution of 1.6. With ours, it goes up to 1.9. For AAV5, it starts at 1.5 and goes up to 1.7. The next experiment we did is kind of a more use case type scenario that I think a lot of people will see. In this case, we have AAV8 material purified from feedstock. So we purified it using an affinity chromatography resin, and then we took that elution and we put it on our QStat running a plateau gradient, as we're calling it, using our proprietary buffer. You can see with this plateau gradient on the left here, we have the fluorescence data, where we have a very large empty peak and a smaller full peak. We also looked at the UV 260 and 280 data, and you can see on the right, the blue is the 280 and the orange is the 260, and above each peak is a 260 and 280 ratio. So using this, we can confirm that the first peak is empty capsids, the second peak is full capsids, and it gives us a proportion of about 85% empty, 15% full, which is pretty standard ratio for a hex cell-based, affinity elution. Now, as far as what exactly our buffer is, again, I can't tell you, but we do have a publication coming out soon, so keep your eyes peeled. All right, and then we're on to the conclusions. So multi-angle light scattering detection showed excellent sensitivity for characterization of AAV using SEC columns with many AAV serotypes. The analysis is highly robust and reproducible, enabling the determination of capsid titer, empty-full ratio, and aggregation content in a single analysis. Anion exchange chromatography with the TSK gel QStat column allows for baseline separation of empty and full capsids. AEX shows linear response in empty and full composition versus fluorescence response. And AEX offers simple setup, high throughput method to effectively separate empty and full capsids for multiple serotypes. The next step that we're working on right now is merging our AEX QStat method with the multi-angle light scattering to combine really good separation with really good detection to give you a really robust AAV characterization suite.